Worked with another person, Christine Volwinkel. I probably said her last name wrong. I'll cut that on the video. I always do that. Some videos it takes me like 3 hours to edit because I've like put so many crazy things in there. Anyway, so another group I work with works on acute lung injury. And acute lung injury is terrible. You know, I got into pulmonary division. This is so sad. I thought when I got into pulmonary that it was blood vessels, but you know, it's lungs. I was like, cool, you guys work on lungs? Great. Me being the PhD. But acute lung injury is horrible. That, you know, the pulmonary division is also critical care, that if you can't breathe, you're going to die, right? And you don't have time to wait. You know, you'll bleed out before if you're not breathing, you're going to die quicker than if you would bleed out. And acute lung injury, there's all kinds of things that happen to little air sacs. But a big thing that happens is you get edema in the lungs, and it basically fills up with this protein-rich fluid. I work with this group, and I'll kind of hurry up here, that believes this gene, or lots of groups believe Glycogen Synthase Kinase 3 beta is involved in this clearance. It's involved in a lot of energy metabolism, a lot of different functions. But what they do is they believe that it has a detrimental role during acute respiratory distress because it inhibits the alveolar epithelial protein transport, and you can't clear the lungs, right? So what they want to do is somehow be able to regulate this after you have acute lung injury, okay? And again, they came to me, hey, we know this gene's important. We have no idea what it does. So I use the same data sets, right? You know, the Dallas one and the one from Novartis. Again, I can look at the expression of this gene in our big data set, and you can see it's varied. So I have a good sense that we're going to find a lot of good correlations. And again, when we do this, we correlate in both gene sets. Again, it's just a Pearson correlation, very simple statistical test. But given the fact that I'm using 2 data sets, I can filter out those false positives and end up with 120 genes that were correlated to both. Here are the top 15 genes correlated to this GSK3B in both data sets, and here are the R values and what they were ranked. And you can take the average rank and kind of rank them this way. There's a ton of cool stuff in here. I wish I could talk about it, but you know, there's this dead protein. It's all about apoptosis. Maybe you can inhibit that. You know, PIK3C3, some of these other ones. But what I want to talk to you about, it'll be this one. It's ELAVL-1. Here's what happens. I find out all these cool things about their system, and I go, look at all this stuff, and they go, we only got this much money, Mike. we can only go for one thing. So what I'm going to tell you is they basically validate this ELAVL-1. Ooh, it's MAD4. Here's a top network. Again, I used IPA to figure out, you know, are any of these genes related regardless of whether in the same pathway or not? Is there any information about are they related in the literature? And sure enough, here's GSK3B here. And basically all of these things, if it was red, it was positively correlated to this expression. If it was green, it was negatively correlated. And all the R values are given below it. The odds of getting all of this by random chance is 1 times 10 to the negative 68th. Again, that's the same odds of, yeah?
I was wondering if the literature refers to utilizing like RNA-seq data.
What's that? Yeah, totally. You can use RNA-seq data.
I was wondering then, how do you kind of compromise with the idea of like batch effects or the effects of like the environment on RNA?
So you treat each center differently. You do your statistical tests separately. That there's going to be error. The worst thing you could do is I could take all that data from both centers and combine it all and do one statistical test. What you find is error is kind of isolated in different groups. And if I combine, it's kind of like peeing in the pool. Like once you get urine in the pool, you can't get it out. If I combine all those samples, all that error from both studies will kind of leak in and I can't get it out. So what I do is I do my tests separately and I say, okay, this center, you know, has its own error, but I'm going to do I got this list of genes from there. I'm going to do the same test in another one. I get this list. What's common to those two lists? That's how I get around that. But here, I mean, when you're looking at things like a network, like take a look at this. If you were going to try to disrupt this network, where are you going? What do you think? Any ideas? I mean, just common sense, guys. What do you think? What gene? What genes? What about this one? ELAVL-1. Look at this. Look at all the things it's affecting, right? Same way here, CAND1. Affecting a lot of things. These two talk to each other. And here's an interesting thing is this ELAVL-1 is also known to interact with GSK3B, right? That's a lot of evidence right there. So basically, this was like, ding, 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 ding. So my collaborator was like, let's look at that. And again, basically, this is a protein. It's also known as human antigen R, but it'll bind REA elements in messenger RNA and basically stabilize that. Lots of gene has these ARE elements in their messenger RNA. This has a potential to affect lots and lots of genes. I guess what I'm getting at is, if I'm going to affect a system, I want to affect something that affects lots of things in my system, right? Like it would do me no good. Why would I want to go concentrate on this gene when it's affected by this and it affects all these other ones as well? What this is doing is allowing you to get the cliff notes before you even get in your system, and now you know who to go after. I do another lecture where I turn genes into criminals, and you can go see that on YouTube. So they basically investigated, is ELAVL-1 associated or connected to GSK3B? And again, when we basically take this in a cell line, if we use mRNA or if we silence the ELAVL-1 mRNA, the expression of GSK3B goes down significantly, right? And this is at the protein level as well. So again, we took transcript information, now we're at the protein level. Okay, well, what about the opposite? What happens when you stabilize or overexpress ELAVL-1? Basically, they use this chemical to stabilize the message, and after 2 hours, you can see that the expression of GSK3B is significantly higher. Okay. Now, does ELAVL-1, what about just straight up injury? When you actually look at injury, so here's the expression of ELAVL-1, the change in an mRNA. This is our control animals. This is an acid aspiration. So basically, they put the mice out before they do this, obviously, but they'll damage the lungs with acid and then basically measure the tissue afterwards. And you can see an increase here after 3 days, but not so much after 7. And then basically they'll do another kind of injury where they actually put a lot of pressure in the lung. So it's a pressurized injury, and you can see this this protein's going way up, or the mRNA is going way up. This is the pattern of GSK3B in that same kind of setup. It looks almost exactly identical to ELAVL-1, right? And now they have a model. And no one knew this before, that ELAVL-1 is connected to GSK3B. Well, actually, in this particular way, in this particular tissue. And again, we published this, that we started out, we found that GSK3B, we correlated, tried to find its superfriends in Legion of Doom in both 2 different data sets. Once we found a list, we basically got targets, and from those targets, then we experimentally tested that to see if these two were connected. And sure enough, when you decrease one, the other one goes. When you increase one, the other one goes up. And they seem to follow the same pattern and the same types of injuries that we're looking at.